guys, welcome back to the channel. So in this video, I'll be talking about a new topic which is plant biotechnology. So I've already made an introductory video of this subject. So if you haven't checked it out, you may check it out. So let's get started with this video. So we'll first describe some of the current problems and solutions with the demands. So as you can see right on your screen, so some of the current problems as in general we face are the population rise, increase of diseases, malnutrition, and learning the secrets of nature. And some of the solutions that can be derived out of that is designing a novel plants, superior in traits, tissue culture techniques, adding value to our natural crops, and some of the demand that is increasing day by day, which is the plant with higher yield, resistance to diseases, more nutrients, and need of more natural products-based medicines. So these are some of the demands that are getting higher day by day, due to which we are facing certain current problems, and some of the solutions that can be designed out of this is this. So as you can see, some of the solutions here are the designing of novel plants and tissue culture techniques and adding value to our natural crops. So we'll discuss one of the most important technique that comes under biotechnology and which acts as a massive solution for all of the problems that arise in our current world. So we'll be talking about Agrobacterium-mediated gene transfer. So I'll be explaining about this. So this video will come in two parts. So I'll be explaining the Agrobacterium-mediated gene transfer in this video, and in my second video I'll be explaining each and every part of this mediated gene transfer. So let's get started. So what is Agrobacterium firstly? So Agrobacterium is a soil-borne rod-shaped gram-negative bacterium. So it's soil-borne or a soil bacterium, and it is gram-negative in nature. Also, Agrobacterium tumefaciens is a causative agent of crawl-gall disease, an economically important disease of many plants. Also, Agrobacterium rhizogenes, which is involved in induction of hairy root diseases. So Agrobacterium has different sort of species. So one is tumefaciens, another is rhizogenes. Yeah. And also it has more number of species. So we'll be focusing on specifically on Agrobacterium tumefaciens. So Agrobacterium found in rhizosphere, or rhizosphere is the region around the roots of the plant where it normally survives on nutrient release from the plant roots. So we'll be talking about these Agrobacterium tumefaciens, which are generally found around the soil where it can survive with the help of the nutrients released from the plant roots. Moving on with this. So talking about the genetic engineering by Agrobacterium tumefaciens. So definitely, so before I talk about more about Agrobacterium tumefaciens, I'll clear one thing about this thing, that the Agrobacterium tumefaciens acts as a natural genetic engineer. So it does not require any sort of mechanical or in silico techniques for genetic engineering, whereas it can naturally genetically engineer itself. So it can naturally bring changes in itself. So I'll be explaining how it does so. So Agrobacterium tumefaciens can infect the plants at the wound site and cause diseases. So wherever the plant is hurt, or wherever the plant releases certain sort of toxins or chemicals, the bacterium tumefaciens get attracted towards that area. So whenever these tumefaciens see that the plant is wounded and release certain sort of chemicals known as phenolics, so these tumefaciens get attracted to that area and get attracted to that part and get stuck to that. All right. So wounded or damaged plant cells. So wounded or damaged plant cells release chemicals such as sugar and phenolic molecules, which can be acetylstingone. All right. So these Agrobacterium is attracted to the wound site via chemostasis. So this is attracted to the wound site by a process known as chemostasis, in response to the chemicals and attach to the plant cell at the wounded site. So as you can see, so whenever the plant is wounded, so these tumefacines, as you can see, these small tumefacines get attracted to the wound site. So this is a enlarged magnified picture. So alright, so these tumefacines get attracted to the wound site via a method known as chemostasis. Alright, also part of TI plasmid, which we known as the tDNA region. So part of TI plasmid, DNA is actually transferred from the agrobacterium to the plant cells where it becomes integrated to the genome of the plant cells. So what happens in this case is, so this agrobacterium mediated gene transfer. So this agrobacterium tumefacin. So these are small molecules which we known as the agrobacterium, which contain a TI plasmid within themselves. Alright, so this is the Agrobacterium tumefaciens. Alright, so this is the agrobacterium. Alright, so it contains a TI plasmid within itself. So as you can see, this is a circular plasmid. As you can see, same here in the circular plasmid as you can see in this picture as well. So this is the circular region that is present inside the agrobacterium, which we known as the TI plasmid. So TI plasmid also contains a certain region which we known as the TI DNA. So small part of the TI plasmid. So small part of the TI plasmid is known as the tDNA. Alright. So let's say this is the tDNA. So what happens in the entire process is this tDNA gets transferred from the agrobacterium to the plant. So this transfer takes place. So this agrobacterium or this tDNA of the agrobacterium gets transferred to the plant. Alright, so this tDNA of agrobacterium gets transferred to the genome of the plant cells. Alright, so this is what the entire process is all about. So this transfer is not as easy as you think. So this takes a lot of happenings. So this takes an involvement of a lot of genes so that the transfer is completed. So we'll get through the knowledge of whatever happens. So this is the entire process. What happens is the transfer of TI DNA of the TI plasmid of agrobacterium is transferred from the agrobacterium to the plants. Alright. So this tDNA or the tDNA, so now we know what is tDNA. The tDNA is the region of TI plasmid of agrobacterium. Alright. So this tDNA carries genes that encode proteins involved in hormone biosynthesis and biosynthesis of novel plant metabolites called opines and agropines. So this tDNA region, so as you know, so this tDNA region contains a lot of proteins. So these contain different sorts of proteins that are involved in both hormone biosynthesis and novel plant metabolites as well. So these tDNA region contain different sort of proteins which are involved in hormone biosynthesis, which can be auxin and cytokinin biosynthesis, as well as synthesis of plant metabolites called opine and agropines. Alright. Also, this transformed plant cell then synthesizes higher than normal amount of these two hormones. So we'll be studying more about this. Also, overproduction of these enzymes such as auxins and cytokinins cause the infected cell to enlarge and divide more rapidly than normal, leading to uncontrolled cancerous type of growth. So sometimes what happens is, so whatever proteins the tDNA or tDNA stores, the overproduction of certain hormones such as auxin and cytokinin causes uncontrolled growth. So this is the picture of uncontrolled growth in the plant cell when the transfer is over. So when the transfer is over, when the tDNA is in the plant genome, so due to the overproduction of hormones such as auxin and cytokinin, it can act as an oncogenic gene due to the uncontrolled cancerous type of growth. So auxin and cytokinin are named as oncogenic type of genes, which can cause cancerous type of growths and thus ultimately lead to the death of plants. Alright. Also, this growing tumor serves as a sink for phloem transport. Nutrients delivered by phloem are in part used to make opines, which are secreted. Also, bacteria living in the spaces between the plant cells take up the opines and catabolize them. Also, Agrobacterium is thus a natural genetic engineer. Alright, so as I earlier said, that Agrobacterium or Agrobacterium vector is a natural genetic engineer. So it does everything itself. It does not require anyone to bring changes in them. Alright, so this was the entire process that I wanted to convey to all. So this is what happens, that the tDNA of PI plasmid of Agrobacterium gets transferred to the plants, and thereby the activity is moved on. Alright. So when the transfer takes place, due to the presence of different sort of proteins that encode that are present in the tDNA, such as auxins and cytokinins, which are involved in hormone biosynthesis as well as biosynthesis of novel plant metabolites such as opines and agropines. So opines and agropines helps in the growth of the plant. Alright, so the opines and agropines are majorly involved in the growth and the synthesis of major hormones of the plants. Alright, and whereas auxin and cytokinins are oncogenic genes, because as overproduction of auxin or cytokinin gene may lead to cancerous type of growth. Alright, so overproduction is very much harmful for the plants, whereas optimum production is always a good part. So let's just keep this video till here. I'll be explaining the entire process of Agrobacterium mediated gene transfer and every step in detail in my next video. So stay tuned and thank you for watching this video.